And then afterwards, he pursued his PhD training in Rutgers University, New Jersey, in RNA genomics until 2012. So after which, Zhe keep on pursuing postdoc training in Harvard Medical School and Broad Institute of MIT. So he was trained by Kevin Sherberg and Avi Reger in system biology and then computational biology. From 2018, Zhe started his independent research program as assistant professor in Department of Pharmacology, Feinberg School of Medicine. At the same time, I think he also affiliated to Department of Biomedical Engineering, both in the Northwestern University. So his lab actually mainly interested in developing and applying integrated computational and experimental genomics approach to examine the regulation of gene transcription and RNA translation underlying cell fate commitment and oncogenetic processes. His lab mainly focuses on reviewing novel disease therapeutic strategies for precision medicine and immunotherapy. His lab interested mainly included to study regulation of RNA translation and then regulation of alternative polyadenylation, and then also regulatory to study the regulatory circuit in controlling tumor promoting inflammation, which I think we are gonna hear a lot of today. So with his training and different achievement, I think he get numerous awards, which include the Linsager Scholar from Linsager Breast Cancer Foundation. In 2020, he got NIH GMS Maximizing Investigator Research Award, R35. So I think, yeah, without further ado, I think thanks a lot for sharing your science with us today, and then we are looking forward to your talk.
Oh, thanks very much for the trans invitation. I'm glad to share our work. So maybe my topic is kind of different from this seminar series. So my lab is mostly interested in basic gene regulation. Basically, the central dogma of gene expression is pretty fundamental. Although we kind of interested in leveraging these knowledges to study like cancer and cancer immunology, but I think currently we are still at the early stage, like understanding some basic molecular biology pathways in the cell. So my lab basically is interested in developing some integrated computational and experimental approaches to study the steps of gene transcription, especially the transcription termination in the nucleus and also RNA translation in the cytosol. So I think most labs are interested in promoters if you study gene expression, but I think a lot of things unknown is about how the RNA polymerase II actually terminate in the DNA, right? So if you think about the termination, actually is very important. If the termination is not efficient, this POL2 can keep going and then run into downstream gene that will cause like a like fusion genes that's not wanted in the cell, right? So, but I think currently there are a lot of unknown questions about this termination mechanism. So I will kind of try to focus on this. The other aspect we are focusing on is RNA translation in the cytosol. So a lot of genomics work has been done about the promoter, but like how the ribosome actually is got engaged to the RNA and decode these overlinear frames into protein, actually still a lot of open questions. So today I will talk about like two recent work done in our lab. So the first one is develop a deep learning model to identify genome-wide cleavage and coordination sites at the nucleotide resolution, and then how I show how this model can help us to reveal novel genetic mutations causing human disease. The second is to develop computational methods to identify genome-wide translated overlinear frame, and we found a lot of so-called non-coding regions can actually encode functional proteins. So I guess, I mean, a lot of things we do is kind of maybe not familiar with by the experimental biologists. If you have a question, you can ask, right? So you can stop me and ask. So let's talk about the cleavage and pollination. So basically, if we think about the RNA, right? So you have a poly A tail, the 3' end, but how the 3' end is determined, right? So basically, when the pol2 run on the DNA template, and it will actually pause at a certain moment and allow this so-called cleavage and pollination machinery to be recruited around this cleavage site. So this when the cleavage is is down here, and then the poly tail will be added to the mRNA 3' end. So that's basically is essential for the generating a stable RNA, and also essential for the termination of the RNA polymerase II. When this cleavage happens, so this basically they generate a nascent RNA here, and then the exosomal complex will digest this nascent RNA, and then when its digestion is complete, then that will cause this RNA polymerase II conformation change and then release from the DNA template. So this cleavage and pollination process basically is essential for transcription termination and generate functional mRNA, link RNA in the cell. But how this cleavage and pollination complex is recruited is through these cis-regulatory elements around the site. This includes this AUA and one nucleotide variant that bound by CPSF complex. And after this cleavage site, there is U-rich and G-rich element bound by CSTF complex. And then there are some other regulatory elements like a UGUA element that exist in a proportion of the polysite bound by CF1, and some U-rich element bound by FIP1, and downstream can have G-rich element bound by the HRMP proteins. So, importantly, one gene can have multiple polysites. So basically, if we have some kind of consensus motifs like this, then cleavage and pollination can happen. So this site, actually, some of the sites are located in introns, some are located in the 3' terminal exon. By using different sites, then this gene can generate different RNA isoforms. So basically, this called additive pollination. So like the APA in the terminal exon region can generate RNA isoforms with the same coding sequence but different 3' TRs, while the intronic pollination will generate RNA isoforms with different coding sequences also, as well as different 3' TRs. So this additive pollination basically play a very important role during development, oncogenesis, and disease, and also in immune system. It's well known that there are some immune genes like immunoglobulin. So they have this intronic pollination can generate different protein sequence that have different C-terminus domain. And this C-terminus domain sequence of the protein basically can regulate this protein localization to the membrane or to be secreted. And so this APA basically play a very important role, just like additive splicing, regulate different aspects of the gene expression. So in our lab, so basically we kind of try to understand how the optimization of these polysites happen in the human chromosome. So if we think about the motifs that are important for the cleavage and pollination, right? You can have AAUA, AUU, AAA, right? So multiple poly signals that can randomly happen across the human gene. But if you think about it during the evolution, this existence of the signal should be co-evolutionarily optimized with gene structure. So if there is a random uncontrolled cleavage and pollination happen at this site, then that will cause improper transcription termination and generate a truncated protein, right? So that's something our cell doesn't want. You really want to generate this kind of we want the exact isoform, right? Like this the expressed site, right? But not use those random sites. how to we try to understand, right? So basically, how the distribution of these signals optimized across the genome. So to address this, we aimed to develop a deep learning model to scan the human chromosome and identify putative polysaccharides at a single nucleotide resolution. So basically, as I mentioned, right? So this pollination happens through multiple motifs, right? Relative distance and of those motifs are important. Whether we can develop a deep learning model just to use the sequence as the input, we can scan this gene and get this putative polyacid at a single nucleotide resolution. And then we can ask a lot of different questions. So basically to say where the putative sites are located, what combination of the cis-regulated elements control this pollination activity, and how the local gene structure determine the site expression. For example, if it is an intron, there is this competition with splicing, right? If splicing happens, then this intron will be removed. This cleavage and pollination will not have time to happen. So to address these questions, so basically we need to have a positive sequence. So what are the real true true sequence, right, of cleavage and pollination? So to do that, so basically we analyze the data called the 3' reads. So basically that developed during my PhD. So the idea is like we take the whole total RNA from the cell and then use oligo-DT column to select the poly-tailed RNA and then do this fragmentation. After the fragmentation, we use a long oligo-DT to select this poly-tailed fragment and then do on-beat RNase digestion and do the adapter ligation. Then we map the sequencing reads to the reference genome and the transcriptome. And then basically those read peaks represent the cleavage and pollination site, right? So so using this data, so basically we can map the genome-wide cleavage and pollination site at a single nucleotide resolution. So basically there are some A's is not from genome sequence, but from the cleavage and pollination. Then and using this reads, we can quantify the site expression, different isoform expression in the cell. So we analyzed like 1.5 billion 3' reads through 19 cell and tissue types and get this near 300 million polyacid supporting reads. And then basically using these reads to identify the genome-wide polyacid. So this is about 110,000 polyacid in the human protein coding genes. So if we think about the sequence around this cleavage and pollination site, so we can see like A-rich motif upstream of this polyacid and U-rich around this cleavage site that represents the cleavage and pollination signal. So these complexes bind to these motifs. So there are 44% A use this poly signal AUA, 15% A use AUUA, and 41% use other pass variants. So basically, we have a basically true positive sequence of the poly sites, right? So those are the positive sequence. Basically, we use 240 nucleotide sequence around the polyacid as a positive sequence, right? And then basically we can generate some random genome sequence or randomized sequence as a negative sequence. And then we develop a deep learning model to classify this positive and negative sequence, right? So this is like using deep learning. So we can have this 240 nucleotide sequence and also use one hot embedded encoded sequence, right? To represent this nucleotide sequence. And then go to this first convolution layer, like to capture the interactions among the hexamers of this sequence. And then the second layer of this bidirectional LSTM, right, to capture the dynamic interaction of the motifs. And then basically have the dense output layer. So basically, we designed this algorithm quite unique. So there are some algorithms developed just basically to this classification. So basically, to classify this positive and negative sequence, like 0 and 1, but we designed this algorithm kind of unique, not only to get a probability, but also get a cleavage probability in the middle 50 nucleotide region of this 240 nucleotide sequence. So this is very important because combine these two information, we can actually achieve the single nucleotide resolution prediction of the cleavage and pollination site across the human genomes. So basically, we can for one gene, for example, we can get like a 200, like we can apply our model, take every like a 240 nucleotide sequence and move one nucleotide per step, right, to calculate this classification probability and get the cleavage probability. So if we think about this is actually the real poly site here, right, from the three-prime rays. So basically we can get a kind of broader region about like a 15 nucleotide region there to be positive to say, oh, this is the poly site. But we don't know where exactly the cleavage is, right? But by combining with this kind of cleavage vector, so we can actually, by averaging them out, we can achieve the singular nucleotide resolution prediction of this cleavage and pollination. That's corresponding well to this observed. So this, our model basically is the first one, right? So basically it can do this. Because of the unique design of a deep learning model, we can scan the human chromosome, get those putative site at a single nucleotide resolution. And also, we basically develop another deep learning model that's kind of a smaller model just to calculate the polyacid strength. So basically, if you think about the terminal exonic site, some sites are expression level high, some expression level low. That's because of the like a motif variance, right? Some of the motifs are stronger, and you have a stronger binding affinity to the factors. Some motifs are weaker, they have a weaker activity. So basically, we build a baby model. So basically, using the input sequence is a 240 nucleotide sequence, and using similar approach. So basically, to predict this sequence strength, basically side strength score. And then it kind of do well, can classify this highly versus lowly expressed site with AUROC of 0.91. So so then by leveraging these deep learning models, we can actually ask many important biological questions. So the first one is like, what are the motifs important for the cleavage and pollination, right? So basically, we can leverage our model systematically mutate the hexmers in the sequence and replace that to some random nucleotides, right? So if the hexmer is important, then the motif disruption will decrease this probability to say, oh, this sequence is a polyacid, right? If the hexmer is important, then the predicted strength or the probability will decrease. So by systematic disruption of these hexmers in this positive polyacid sequence, we can actually capture these known important motifs in the position-specific manner. So basically, for example, this path AAUA and a variant, we can see exactly its maximum importance score is about 20 nucleotide upstream of the polyacid. And this UGUA motif actually is important when it's located about like a -30 to -90 region. And the U-rich motifs important around the cleavage site. And the G-rich motif located like downstream of this U-rich site. That's corresponding well to the known features of this motif existence and the function in the cell. And also we can use this to study the crosstalk among the motifs. For example, we can fix this AAUA and mutate these UGUA motifs around this site, right? So basically, if the distance is optimal, then this disruption of UGUA will cause a more drastic decrease of the predicted probability. So basically, we can see this UGUA motif is optimal when it's located about 15 nucleotide upstream of AAUA. So you can see this kind of distribution actually corresponding well to the experiments. So there is a paper from Sheng Shen's lab from UC Ireland. So basically they did a report that actually show this UGUA is optimal when it's located like about like 18 or 15 nucleotide upstream of this AAUA. So this kind of shows the power of deep learning, right? So basically we can use this sequence, right, just as an input, and then we can learn the motif grammar, especially the interaction of these motifs in a position-specific manner. So basically, here we can use in this approach to show the optimal configuration of the polyacid, so the relative distance position of different motifs. And also we can apply our model to scan the human chromosome, right? So get this putative polyacid at a single nucleotide resolution. And then we can get the expressed side through this 3' data, right? So basically we can see very small fraction actually are expressed. Vast majority actually are not expressed based on this 3' data. So if we think about the putative side, right, randomly occurring site, polyacid in the human genome, so vast majority are in the intron. Near like 90% actually are in the intron. So a small fraction located in the exon. So, but if you think about the fraction of them are expressed, like only about like less than 2% of the intronic sites are expressed. But like near 40% of the terminal exonic polyacids are expressed. But like a 10%, this is 5% something of the terminal extended region of the exon sites are expressed. So why this intronic polyacid are suppressed, right? They are not expressed. That's because of the competition with splicing, right? So basically, when the splicing happens, this intron will be removed. Then this intronic pollination will not happen, right? So basically, this intron already been removed. So then we basically next study the crosstalk between this intronic pollination and the splicing. So based on the gene structure, we classify this intronic site into two. One is a composite intronic site. The other is called a skipped intronic site. There is a splicing between this 5' splicide and the polyacid. Here, there is no splicing. So we can see for the composite sites, so when the expressed sites tend to be stronger, right? So it kind of makes sense, right? If you have a stronger motif, then it will have better binding and factors are more efficiently recruited, then the site will be expressed. Then we next is control the site strength. Basically, we randomly selected some of these sites and control, right? Basically, they have similar strength, and then say why some are expressed, some are not. So we can see expressed sites tend to have weaker 5' as upstream 5' splice site, and the distance to the 5' splice site are shorter, and actually they're located closer to the 5' splice site, and the intron size actually is bigger. So this intronic pollination more likely happen when it's actually have weaker splicing environment with larger upstream downstream axons, weaker splice site, also in the last intron. So basically, last intron is special. So if you think about the splicing efficiency of different introns, the last one actually is lower than upstream because the axon definition, this last thing, so basically this 5' splice site need to recognize this 3' splice site to splice out the last intron. It's different from the upstream axons because upstream axon is through the recognition of 3' splice site to the 5'. So this is called axon definition. The axon are smaller, so it's more efficient. But the last intron that is special. So this intron is very big. So splicing efficiency is lower. So we basically using this parameter can build a logistic regression model and classify this used versus unused site with AURC of 0.91. And for this, for those skipped intronic sites, so there is an upstream splicing, and here is another splicing. Then there is a competition between this splicing and this splicing. There's the same 5' splice site, but different 3' splice site. We show this intronic site tend to be used when they have higher, stronger like upstream splicing efficiency, but a weaker downstream splicing efficiency, and also when it's located in the last intron. So the next we can actually also apply our model to study genetic variants causing human disease, right? So basically, it's known for many years, disruption of this cleavage and pollination signal can cause human disease. For example, so in cancer, so basically this mutation of this AAUA can decrease the cleavage and pollination efficiency, and then the POL2 will keep going, and then the efficiency to generate mRNA and protein or are drastically decreased, right? So basically, these papers show that this mutation of this AUA uh, uh, of TP53 can cause cancer progression. We can apply our model, right? So the model basically to say whether a mutation can cause the decreased cleavage and pollination activity. For example, the SNP I showed before of TP53. But we can unbiasedly identify all the possible motifs that can either disrupt the poly signal or create a de novo poly signal. So these are the ClinVar variants. So this is a disease-associated variant. So about half strengthen the polyacid, some weaken the polyacid. Many actually have unknown significance. So basically, those variants basically seem to link to human disease, but it's unknown why, right? So we suspect to say, oh, the activity on pollination maybe is the causal reason. So, for example, this gene is a plankton gene. So mutation of this gene is associated with muscle dystrophy. So we can see there is a ClinVar variant. Basically, it's a missense mutation, but if we think about the coding function, it's a missense. Actually, it's create a de novo polyacid here, create de novo AUA here. So if this polyacid is used, then this gene will generate RNA isoform without the stop codon, right? So basically, the cleavage and pollination will happen here. So this RNA will be degraded by non-stop decay, right? So currently, we are going to lock in this variant in the cell and then see their functional importance. So this is the first part of my talk. So basically, we show by developing deep learning models, so basically we can uh, uh, study the cleavage and pollination in some innovative ways. So basically, we use the model to quantify the position-specific motif importance, analyze putative site distribution usage, and also assess the impact of genetic variants on APA dynamics. Okay, so this is the first part. Okay, maybe I should just continue, right? To the second part. Second part is about RNA translation. So if we talk about RNA translation, right? So basically, maybe we can first think about the transcript types that are annotated by the human genome. So right, if you go to any database nowadays to say what are the transcripts, right, in the genome. So basically, generally they are classified into four types, right? The first type is mRNAs. This is basically those are the protein coding genes. And then long non-coding RNAs, right? So especially with advance of deep sequencing, people sequence more types of cells and found there's so many, right, long non-coding RNAs. The number actually is bigger than the mRNAs. And there's pseudogenes, right, and a small non-coding RNA. But during the history, right, so basically it has been a challenge to separate mRNA and link RNA, right? How can we say, okay, if we found a new transcript in the cell to say, oh, this is a long non-coding RNA, but not an mRNA, right? So there are two common approach, right? So one approach is to use a cutoff, like 100 amino acid. So basically to say, well, one transcript, if they have, it has a oberylin frame longer than 100 amino acid, then it's a coding transcript, right? It's coding, right? If there's nothing longer, then it's non-coding, right? And also more popular approach is to develop some computational method to calculate the coding potentials of transcripts. So basically, there's some algorithm developed. So basically, to train a machine learning model to say whether this transcript is coding or non-coding, right? So the common way to do this is like you have positive protein coding genes, right? Is a annotated mRNA sequence that with a medium length of 500 amino acid, and that's evolutionarily conserved. That's a positive sequence, right? And then you have some link RNA as a negative sequence, and then try machine learning model. But those models has mutations. It can identify the positive coding sequence coding genes, but actually can ignore those stably folded microproteins our species-specific proteins, right? So basically, the preassumption may have some problem, right? So basically, is to say, okay, if something not long, not conserved, then it's not functional protein. Maybe this may not be right, right? And it's known during the history, there are many functional macroproteins like RPL41, SLN, and APLA, right? They're shorter than 60 amino acid, actually. Some men, if you think about RefC database, actually over 200 macroproteins actually are annotated. So, but how to address this question, right? So we basically recently try to address these questions. So how can we say something is coding or non-coding, right? But if we think about to address this issue is first we need to identify what are the translated overleaning frames, right, across these transcripts. So to address that, so basically we need to go to the fundamentals of RNA translation. So basically, we know the 40S ribosome recognize the 5' cap structure and scan until it meets the start codon ATG and the one nucleotide variant at that site, and then 60S will join forming this 80S ribosome, and then move 3 nucleotide per step, basically 1 codon per step to decode this overleaning frame into protein. So the technology we use to study RNA translation is called ribosome profiling. So basically that's invented by Nick Ingoglia and Jonas Weissman. So the idea is like they use RNase-1 to digest the cell lysate and then isolate this ADS ribosome complex and recover this RNA fragment footprints for sequencing. So it has been widely used for study different aspects of RNA translation. So if when I was a postdoc, so basically I analyzed this ribosome profiling data. So if we do it carefully, so basically we can see there is a clear 3 nucleotide periodicity of the redistribution starting from the start codon and at the stop codon, right? And if we zoom in to specific transcript, actually we can clearly see this 3 nucleotide periodicity for individual codons that representing this ADS ribosome moving this 1 codon per step to decode this overleaning frame into protein. So basically this ribosome profiling technology is very powerful, right? So basically it reviews this active translation at the single nucleotide resolution. So then based on this 3 nucleotide periodicity, we developed a machine learning model based called RibRF to identify the genome-wide translated overleaning frames. So how the algorithm works is like we first select this candidate RFs with a start codon ATG and a 1 nucleotide variant and a stop codon, and then get the expressed ones. And for the expressed ones, we basically use to see whether there is a 3 nucleotide periodicity across these overleaning frames. We use the fraction of the reads in the first nucleotides of the codons and the percentage of maximum entropy to measure the uniformness of this distribution across the overleaning frame. We can see for the canonical RFs of mRNAs, right? So we can see the high fraction of the reads in the first nucleotide and the uniformness of the reads across the ORFs. But for some of the long non-coding RNA and far-primitive RFs called URFs, they actually also show similar patterns. Basically, those RFs are actively translated. So we can achieve high accuracy and sensitivity with this AUROC value greater than 0.9. So recently, we basically applied our approach to nearly all the published ribosome profiling data from yeast to humans. So basically, we then annotated this translated RFs of the mRNA, link RNA, pseudogenes annotated by the GENCODE database and the different also in other species. So we can see is for the mammalian translaton and also mouse, their translaton is highly complex. It's much more complex than yeast, right? So if you consider human, so the annotated protein only RF is only account for about 40%. There are about 60% of the genes show translation in 5'-TR, 20% show translation in 3'-TR, and encode those tens of thousands of these non-canonical RFs called URFs. That's the overleaning frame in 5'-TR, overlapping URF, internal RF, downstream RF in 3'-TR, and the long non-coding RNA ARFs and the pseudogene ARFs, right? This is called non-canonical ARFs. So now in recent years, right, and this has become a new research field, right? So my paper actually was one of the first to comprehensively annotate this translaton, but by identifying those ARFs, then we can study their function, right, in a cell. So in the history, people focused on this regulatory function of these UARFs in controlling the downstream ARFs. But now, in recent years, people kind of care about whether the peptides generated by these non-canonical ARFs actually can be functional. Right? So if you think about the protein function of these ARFs, then we need to think about its length, right? So if something is long, then it's probably likely to be stably folded and it can have function, right? But if something is very, very short, probably they're degraded, right? They don't have any function. If you think about the non-canonical ARFs, their medium length is 16 amino acid. For something about 16 amino acid, probably not functional, right? They're too short. But there are outliers. Here I draw the line, this 100 amino acid. So there are actually a good number of these non-canonical ARFs actually can generate peptides longer than 100 amino acid. For example, this is a UARF peptide that encoded by this 5' TR. We can see there is a ARF that's 166 amino acids. They are not too short. So this is now is a new field, right? In the past years, there are a few important papers published. So this one is like we collaborate with Todd Golub's lab at the Broad Institute. So they took our list of these ARFs identified in cells and then did a systematic overexpression. Hundreds of these link RNA included these peptides, and found many of them actually can be stably expressed in the cell. And they also did CRISPR screening to show that knocking out those ARFs impact cell survival. And this work is from Jonas Weissman's lab. They invented a ribosome profiling. Basically, used a similar approach and reached the same conclusion. So basically, many of these UARFs encoded and link RNA encoded peptides actually may be stably expressed in the cell and can have biological function. And this work is this is my postdoc's mentor Aviv Regiv and collaborate with Cassie Wu. Cassie is a cancer vaccine person, right? So basically, they did a lot of mass spec of this MHC bond, this immunopeptide, right? So and they have this mass spec data, and then use our software to search, right? To search this non-canonical peptides, and found thousands of these non-canonical peptides encoded by 5' TR, and link RNA actually are presented by MHC, right? So they can function as antigens for disease therapy. And this is another work from Jonathan Yedell's lab, like from NIH, and collaborate with Claude Pro. So basically, they also use this cellular proteome mass spec data and immunopeptide data and search this non-canonical ARF database and found that it seems like these non-canonical proteins either to be found in the cellular proteome or like found in the immunopeptide. So the emergent theme is like there are some of these non-canonical ARFs encoded peptides are stable functional, but many of them probably are quickly degraded and they don't have function in the cell. But they can function as antigens for immunotherapy. So recently in our labs, we did work. So basically, we ask us a question. So how can we computationally distinguish these non-canonical ARFs encoding stable peptides versus quickly degraded byproducts? So which non-canonical ARFs are likely to encode these functional stable proteins? To do that, so basically we did some literature mining. So basically we collected some stable macroproteins that studied in the history. So basically there are near 300 annotated by RefC, right? They have well-characterized function. And also there are 70 something from this John Plessner and John Golub's work. And this is Jonas Weissman's work. So basically, these are the positive macroprotein. They're stable in the cell. That's all shorter than 100 amino acid, right? They're shorter than 100 amino acid. So we call it macroprotein. Meanwhile, we also collected some undetectable macroproteins from epitopic expression. This is from collaboration with Todd Golub. So basically, they did overexpression of these macroproteins and actually found many actually undetectable. Why they are undetectable are likely to be degraded, right? So they're possibly unstable. So we want to study like what's the difference between those stable and undetectable macroproteins. So first we check its conservation, right? So basically this is the people generally do, right? To say, oh, something conserved across mammals, then they are functional protein, right? If something not conserved, probably are not functional. But we basically we show those stable ones indeed is more have higher follow CSF score that measure the conservation levels across the mammal. But if we divide these peptides into three groups based on their conservation level, they actually show similar length or longer than undetectable ones. So it looks like from this plot actually kind of imply to say that maybe the conservation doesn't matter, right? So basically, if something longer than 60 amino acid, it start to be stable, right? But something shorter, maybe it's unstable. So then we ask the question why people originally use 100 amino acid, right? To say, okay, this is a cutoff to to define functional protein, right? There are some pre-assumptions. One is like long peptides are likely to fold it into secondary structure and they're stable. Something is too short is likely to be not likely to be folded, then it's not stable, right? And the other assumption is like long oberlinium frame would not emerge by chance during evolution. So if you think about it, you have an ATG start codon and have one random nucleotide after the ATG, then we will get some random stop codon and the peptide tend to be short. So if you consider those stable macroproteins, the transcript length is about 1 kb. At 2% FDR of this expected random sequence is about 70 amino acids. So basically, a lot of those non-canonical peptides, stable macroproteins, actually longer than expected by chance if you think about their transcripts are shorter. So there is a correlation between the peptide length and the transcript length. But for many of these annotated link RNAs that tend to be short, the encoded ORF lengths, of course, are short, right? But that doesn't necessarily mean they're random, right? So we show they are not random. So and also this stable macroproteins tend to have an encoded protein domain. So then based on this conservation length FDR and the encoded domain, we developed a logistic regression model called a PEP score that calculated the probability an ORF encodes a stable peptide. So basically our PEP score can effectively classify those stable versus undetectable link RNA annotated encoded macroproteins. And there is RefC defined macroproteins also have high PEP score. And importantly, like we found this off length FDR show higher contribution to this classifier than this conservation. So this shows that actually the conservation may not be so important, but the length actually is important. Then we apply our logistic regression model to the genome-wide ORFs we identify, right? So basically, this is overall distribution of this PEP score of these non-canonical ORFs we identify. So there are about 13% actually have higher PEP score than 0.6, but 80% actually have low PEP score smaller than 0.3. It means that vast majority of these non-canonical peptides probably are degraded. They are not stable, but there's a small fraction of those have high PEP score. Those non-canonical ORFs are from the 5' TR, the URFs, overlapping URFs, internal ORFs, 3' TR is downstream ORFs, link RNA pseudogenes. The median length is about 90 amino acid. And importantly is like the poorly conserved across mammals with this negative follow CSF score. So our analysis suggests to say, actually, the length matters. Conservation doesn't matter so much. Probably there exists a large number of these species-specific young proteins. They may be human-specific, mouse-specific. They are not conserved, but they are functional, stable in the cell. So we then did some validation. Basically, we use this URF encoded this peptides. We fuse this URF sequence with the same 5' TR, 3' TR with a flag tag, and then use the immunostaining experiment to show whether we can detect these peptides in the cell. Some are stable, right? Like this one is pretty stable. Some are not so stable. Some are like empty vector. So basically, we can see them at all. Then we use GFP as a control, so we can basically show some of this URF encoded peptides are pretty stable, some not so stable. Then we basically treated the cell with some proteasome lysozyme inhibitor, try to see whether we can rescue the expression. We can stabilize some of these peptides with this longer lengths, but not these very short ones. So we think basically for those very short ones, like generally shorter than 40 amino acid, then they can be degraded by protease, cellular protease. There are tens of proteases in the cell, right? They don't need the proteasome lysozyme pathway to fragment them for degradation. They're just degraded by protease. But some of these like moderate length ones, they can be stabilized upon this proteasome lysozyme inhibition. Then we follow up with some of the stable macroproteins. So for example, this one, so basically is a URF peptide encoded by 5' TR of this mRNA. So mutation of we basically did a full-length transcript overexpression by tagging both of this URF and a MERF with a flag HA tag. This is endogenous transcript sequence. So we can see mutation of this URF start codon will abolish this URF peptide expression and enhance this MERF protein expression. And we're using immunostaining to show that this URF peptide is localized to mitochondrial outer membrane. The twofold overexpression will slow down cell proliferation and damage mitochondrial outer membrane potential. So we think this URF peptide is a mitochondrial membrane protein, regulate cell proliferation and mitochondrial membrane potential. So there is another non-canonical peptide is an internal RF. So basically this is an AUG downstream of this canonical AUG. So this is also about 80 amino acid long. So basically using co-IP mass spec experiment, we show that this peptide interact with this mitochondrial processing peptase. And the immunostaining experiment show this peptide is localized to the mitochondrial matrix. And that there is actually this peptase can cleave here to generate like a C-terminus 11-mer translocate to the cell membrane. And this N-terminus will stay in the mitochondrial matrix. So like twofold overexpression of this will activate some development gene and suppress some immune response gene. So in summary, so basically we think actually, so there are pervasive active translation, right, generate these non-canonical proteins across different RNA types, like including mRNA, link RNA. So, but the stable macroprotein can be generated by the 5' TR, 3' TR overlapping coding regions and this annotated long non-coding RNAs in the cell. So basically from our data, we suggest to say, oh, the long ones, typically longer than 60 amino acid, are likely to be stable, may have biological function. But the short ones that are degraded, but they can function as antigens for immunotherapy. So I think this is actually a conventional approach to create a lot of these species-specific proteins, right? So if you think about the human, mouse, Drosophila, or different species in the planet, right? So basically they can use the existing mRNA structure or the enhancer, right? So basically to generate this kind of so-called young transcripts that will basically generate these species-specific proteins. So finally, I would like to present to say, oh, the functional protein is not contradictory to the RNA function, right? So when we present this work, there are always arguments from the link RNA field, right? People to say, oh, this link RNA is known to have some RNA function in the nucleus, right? But I think it's not contradictory to their translation in the cytosol, right? It's just a matter of the localization of the RNA. So an RNA can have be translated into functional protein in the nucleus and also can have some other functions in the translation in the cytosol, but it can have other regulatory function on the chromatin in the nucleus, right? So they are not contradictory to each other. So finally, so basically, here we presented two pieces of work. So the first is like we developed a deep learning model to identify genome-wide genome-wide cleavage and pollination site at nucleotide resolution. The second part we developed computational methods to identify genome-wide translated ORFs and review stable non-canonical peptides with potential functions. So finally, I would like to thank my lab members. So Emily did the deep learning work, and Haiwan and Qianru did the RNA translation work, and my funding sources. Okay, thank you very much.
Well, thank you very much. It's, quite a new field for actually immunology people. I think I learned a lot about RNA biology. So we can open the floor and then start the question. If anyone have question, please raise your hands or put the question in the chat box. I think I'm gonna to Chuanxue, you have a question or you want to ask a question? Yes. Okay, go ahead.
Yeah, I just want to say hi to teacher. That's
Oh, oh,
hi teacher. I'm
yeah, yeah,
Chunxue.
yeah. We are.
Teacher is a role
PhD
model
classmate.
for all students from UMD India back then. We
So
learned I think we learned a lot from teacher.
where are you now?
Yeah, I'm glad to hear your talk. I have a question about mRNA. I'm not an expert in this area, but now I'm working in the field of siRNA. We are trying to develop drugs for many kinds of disease. But one interesting thing we found, even by targeting different region of mRNA, if you design a lot of siRNA targeting different region, you'll see different potency. Some is very potent, some is very weak. So I'm wondering if you have any input on this, which can help, for example, people in this area, they can by either machine learning or other method can predict which is for example, the hot area, which we can get very good potent siRNA that can save a lot of expense and time for people in the field.
Yeah. I mean, yeah. So I think there are some tools, right? Already try
Yes.
to do that, right? It's related to RNA structure and things like that. If something is structured, then sRNA are less likely to be bound, right? And like a microRNA targeting, like Dave Patel, they
Yes.
developed target scan, right? So basically to see the efficiency of microRNA targeting, yeah, if you have enough data of the like number of the sRNA and then the targeted mRNA, suppose people can actually develop some deep learning model, right, to predict the efficiency of the targeting, maybe classify some high efficiency sRNA versus lowly efficient ones. Yeah, I think it's all possible. Just
Okay.
need to find some people to work on this, right?
Yeah, thank you. Do you think is there some region in MRI, for example, they are more active than other area, while other area is like kind of like a hibernate, which even you don't have access to that area of MRI at all.
Yeah, usually I'm not so familiar. I mean, usually people target circumTR, right? Yeah,
Yes,
I
yes, that's true.
yeah. Maybe the reason is because circumTR is AU rich, is less structured. I'm not sure. Yeah.
Okay, I will learn from you later after. You
Yeah.
know.
Okay. Glad to
Thank
hear
you
from
very
you.
much.
Okay, see you.
Thank you.
Okay, so I think I have one question to actually two actually. So one is from the deep learning, you actually can precisely predict the poly A site on any kind of genes. I'm wondering if you actually ever can execute experiment to verify the poly A site. If it's only single nuclear, you can precise to the single nuclear site.
Yeah, I mean, yeah, so validation, yeah. So basically we did a lot of a reviewer asked us to did a lot of validation. So we first of all we will train the model, right? So basically we split this whole set into like training, testing, and validation. So the testing set it was not included in the training, right? So the performance of a model is always evaluated by this independent testing data. So it haven't been seen by the model during the training. This is one validation, right? The other validation is like we did a lot of uh, reporter, like people already did, like just called using this massive parallel reporter assay to overexpress like tens of thousands of this polysequencing in the cell, right? And then basically you did quantify the expression to see what are the elements important for cleavage inflammation. We reanalyze that data and to show our model can recapture the cleavage inflammation sites that is from this synthetic RNA. We did that in the paper.
But with the report assay, you basically use the mutation connected with the luciferase. Is that what the report you use?
The reporter is it's yeah the coding sequence is the same, right? They're just a randomized sequence around the polyaside.
True, but I think your prediction through the deep learning is you can specify a single nucleotide for the poly side, right?
Yeah, yeah. So
But
basically,
how you actually generate this mutation in terms of to actually validate its function?
it's random. Nowadays there is a technology called a massive parallel assay. So basically, we can randomly synthesize this like tens of thousands of
But
this sequences, and then you do experiment, right? So basically, you perform the like third prime raise or something, right? To basically quantify each of these cleavage events of this synthetic RNA, and then basically we can quantify the relative expression, right?
yeah,
And
sure.
the
But
cleavage heterogeneity.
if you randomize the sequence, I mean, each species, how many mutation you will obtain. I mean, I will imagine if you randomize it every time, you won't be able to only specify one polyiacid. It probably will be several different amino acid mutation, right?
Yeah, but yeah, I mean, is there published ones kind of saturated mutation? You know, there is like even some kind of I think zero point 0.1 million sequences. We actually analyzed a few cohorts. Like you said, I mean, if we're specifically interested in like some polyaside, maybe we should do some reporter assay, right? Design the wild type sequence and then mutated allele. Is a good idea. If we're particularly interested in one
So
gene,
you haven't done so.
we right, we haven't done, right? Because we did a lot of benchmarking using published data, including this genetics data.
Okay.
People basically did RNA GTAX data, right? So people show these functional SNPs. We can use our model to predict its impact in the right direction. Yeah, I mean the reviewers asked us did a lot of benchmark of
I
this
see. I
and
see.
compare with the published software. Yeah,
I see.
yeah.
My second question related to the microprotein. You say that if it's longer than 60 amino acid, it's actually highly likely to be functioning. But what's the threshold of the microprotein? I mean, the longest will be no more than 100 amino acid. I mean, how are you?
So
Yeah.
yeah, so basically this is not something fixed. So I said 60, I think it's something 60 may worthwhile to look into. Not to say 60 is guaranteed to be functional. It's just like something 60 have a good chance, right? So but if you think about this distribution, right? So below 60, probably vast majority are not stable, but above 60, maybe majority like become stable. So I was just say this maybe if someone want to work on this, maybe you can have a rough idea to start with. But in terms of the cutoff of this, it's pretty big. Some people say 100 amino acid, some people say 200 amino acid, but generally here, because we want to train this machine learning model, we limit it to 100 amino acids. We only focus on these very short ones. But the model we developed is called logistic regression. So basically, it's a linear correlation. So something short, longer, more conserved, then you have a higher score. So not limited by the length. So if it is longer, then you have to get a better score. But in terms of this overall non-canonical proteins, like including the link RNA encoded peptides, some actually can be very long. Like for example, many of those ones, right? 2 to 8 is what is actually maybe 200, 250, right? So this some actually pretty long, over 200 amino acid. But people say this is a link RNA. The reason why they say it's link RNA is because this peptide is not conserved, not conserved. So they have a they have a negative this follow CSF score. So that's related to the algorithm people developed to so-called calculate this coding potential of the transcript, like I discussed in the beginning. So in the link RNA field now, people use this computational method to calculate the coding potential is problematic because their positive sequence, the median length, if you think about the annotated proteins, the median length is about 500 amino acid, and they are conserved, right? They say something about 200 amino acid, 100 amino acid, not conserved, then they are not functional. I mean, that's the intrinsic problem of the existing software. So our work is suggesting to say, is to say that this is probably not right, right? Something kind of longer but not conserved, they can be stable and functional in the cell.
Yeah, but if it's longer and conserved, how
Not conserved, not conserved. Just
yeah,
something long enough, but not conserved. They can be functional.
I know, but how can you, I mean, I guess the point of whether this microprotein is functioning or not is very much depend on its stability.
Right.
How you predicted the stability of the microprotein?
So we developed this so-called PEP score. So basically, you have a higher score, then more likely to be stable, right? So we just give a probability. But if you really want to study them, you really need to do experiment to validate. We just say overall there is a correlation between general stability and our score, right? If we just our score is give a guideline to say, okay, if you really want to work on this, which ones we should start with, right? But really uh, depend on the later experiment, right, to show, okay, indeed they are stable in the cell.
Okay, I see.
Yeah, it's just like people are kind of like prioritize.
I see. Okay. Uh, okay, so thanks. I don't see any further question in the chat box. Anyone have question in the audience? Have question for? Oh, there's a question here from Yan. Are the ORFs in tumor cells more active to express microproteins? What make them different from those in normal cells? Is there any similarity of the short digestive epitopes between species?
Yeah, so nowadays, I mean, there are a few paper published that show some microproteins in actually regulate cancer cell survival. The Yi Wencheng's lab published a few, and John Preston's group also published a few. Yeah, so basically, so the general approach they did is do some CRISPR screening, right? So basically now we have the ARF sequences, then we design guide RNAs, right? A few guide RNA to each ARF, and then do CRISPR screening, and then using some negative guide RNA as a control, then we can see, which knocking out which ARFs can impact tumor cell survival. So what makes them different from the normal cells? I haven't seen anything systematic to say, okay, to compare the microprotein expression in, for example, breast cancer versus normal tissue. So actually, I haven't seen something systematic like that. So the one reason is because the limitation of Raphson profiling data, right? So currently, the data is mostly generated in the cancer cells. I haven't seen anything like systematic to do profiling. Oh, John Presner that did it, like in some brain tumor. brain tumor, they did like a normal and a tumor profiling, and then they see some showed some microproteins have higher expression or unique expression in the tumor. Yeah, but generally I think it's still an open question though. So in terms of species specific of these antigens, I think it's really depend on the MHC, right? Human and mouse are quite different, right? So so like Kathy Wu's lab, they showed, right? So this different MHC can bond like by recognized by different peptide sequences. There is a position where matrix, right, for different kinds of MHC alleles. Yeah, so it really depend on the system you study, right? So yeah, I mean,
Okay.
can be different between human and mouse.
Okay.
Yeah.
Well, if there's no further questions from the audience.
Excuse me,
Okay, well, there's one.
can I ask a question?
Yeah, go
Sure.
ahead. Of course.
Okay. Oh, thank you. Yeah, thank you, Professor Ji. That's really a great talk. I actually requested a protocol from you regarding the RiboSeq, so you give me a lot of good suggestions. So here, I just want to thank you a lot.
Oh, so it works? You tried?
Yeah, that really works because I had some sample with low input, like the mouse or salad. So I try to optimize that one you give me, and that works really well. So
Yeah.
take this
That's
opportunity to thank you. Yeah.
yeah. Thank you. Yeah, I'm glad to hear that. I think that work I didn't present today, but that protocol actually changed a lot of things in the RNA translation field. I mean, because of that work, we actually got got appreciated by field leaders. I sent my protocol to Nick Ingolia. Yeah, he said it works. But many people are suspicious at the beginning. I mean, when we tried to publish that paper, it was so difficult. I mean, the reviewers didn't want to believe it. I mean, they write long stuff just to say, I don't want to believe it. One paper. I mean, in the end, we publish in Genome Research, but it's sometimes it's difficult to
I
do.
think, yeah, that was really for the low, especially for the low input sample, because in our facility, we don't have a very convenient ultra centrifuge here. And also that takes a longer time. So using your protocol, I think it takes less time. That's good for me.
It works well. I mean,
Yeah. So I have a question. Yeah, just to save the time. So I have a question regarding your first talk, the first part. So you mentioned that I think that's it was really striking that the polyadenylation site takes place occurs a lot often in the intronic region. I never know this one because if that's the case, I think the question is regarding the sequence order that takes place between the polyadenylation and the cleavage. So because the transcription is the both polyadenylation and the cleavage site are linked together during transcription. So if the cleavage only happens in the last in the terminal sequence, that means the intronic sequence should be removed first during transcription. So I'm just trying to ask if that's the case, that during transcription the intronic sequence should be removed before the cleavage happens.
So it's splicing, right? Splicing is co-transcriptionally happening. So basically, it's much more efficient than cleavage and pollination. So during transcription, so these splicing factors and the poly factors, they actually got recruited by the POL2. They actually nowadays like Xiangdong Fu's lab, right? So basically they found a lot of RNA binding proteins. Actually, like the splicing factors, they are transcription factors, right? So basically, the chip C they together with POL2, and then they elongate, right? And they start to recognize splice side, poly side, and then basically when then the fact complex is recruited, then basically the splicing and the pollination can happen if there is a if there is a efficient time, right? The efficiency is enough. So yeah, so people did that. I mean, it's co-transcriptionally happening. For the poly A, basically there are some of the, for example, promoter pausing, right? Promoter pausing. I mean, a lot of this cleavage inflammation in the first intron actually happen a lot, is causing this POL2 premature termination. And yeah, so also some of the genes, not every intronic pollination is not is junk. Some of the very important genes, like immunoglobulin gene, they have this kind of intronic pollination regulated during, for example, during this immune cell differentiation or T cell activation, right? So basically to switch this C-terminus domain of the protein, that can be regulatory, make a protein to be secretory or membrane bound. So there are a lot of regulatory function as well. Yeah, so yeah, it's all co-transcriptionally happen. Yeah.
Oh, so somehow, so you think that the intronic polymer deletion site can also regulate the transcription level.
Yeah. So basically, yeah, it's known. Yeah, some of the genes, if you have highly expressed intronic site, this is called alternative pollination, right? So basically, so in different like especially immunology, actually, it's the Christy Mayer at Sloan Kettering, they did a lot of profiling, and the first case, probably the first case of this alternative pollination regulation importance is in immunology. This is immunoglobulin M gene. So basically, there is an intronic polyacid in the last intron. So basically, if this one is used, maybe it's a membrane bound. And if this side is used, then the C terminus of this protein is different. Then this is secreted, right? So basically, there are some genes using this mechanism called alternative pollination, right, to generate different protein isoforms in different cell state. So during development, cancer, any disease, they all can happen.
Okay, so thank you. So you got in your second part, so you mentioned the microprotein. So I'm just wondering, there's a tissue-specific microprotein profile like the